Good morning, students, and today I am going to discuss about the meiosis stage. Okay, the meiosis stages, which are having four different type of stages, the similar with the mitotic, that is prophase and metaphase, anaphase, and telophase. Before that, I am discussing about stages. I am going to discuss about the definition of meiosis. What is definition of meiosis is nothing but while the sex reproduction which are observed the megasporogenesis and microsporogenesis. What is megasporogenesis and microsporogenesis is nothing but the megaspore mother cell which contain two set of chromosomes. This mother cell which producing four daughter cells. This process which is called as meiosis. See, these daughter cells are unidentical with the mother. Okay, at the same time microspore. Okay, I mean in the meiosis process, only in the meiosis process the only diploid cell is participated, not haploid and triploid. Why? Because in the meiosis we can observe two different type of meiosis 1 and meiosis 2 types. See, what is meiosis 1 is nothing but the mother cell which producing only two daughter cells, that is meiosis 1. Then these two daughter cells again which participating one more meiosis, that is the meiosis 2, which producing four daughter cells. In this type of two meiosis and mitosis stages are present, types are present here. Okay, so this type of meiosis and mitosis process was first discovered by J.B. Farmer and Mood scientist in the year of 1905. Okay, and the meiosis which are help in the process, especially sexual reproduction, not like vegetative and asexual reproduction. Okay, these both asexual and vegetative reproduction, that is belongs to mitosis. Okay, now I'm explaining the facts of meiosis. So the facts of meiosis is nothing but the preceded by interphase, which includes chromosome replication. Whatever two mitotic divisions as meiosis and meiosis 2, the meiosis is also called as reduction division. Why? Because the chromosomes are reduced up to 50%. Okay, then organelle cell is diploid, which means the mother cell is always haploid, not haploid and triploid, which are having two set of chromosome, that is nothing but the mother cell having diploid condition. Then finally four daughter cells are the four which give four daughter cells. These four daughter cells are unidentical with mother, which means which are having haploid, either decrease of chromosome numbers up to 50%. Okay, this is the most fact about meiosis. This is also the fact of meiosis, the produce gametes, which means the meiosis process which are helping only reproduction time. That means at the time of production of whatever male gametes and female gametes, that means which the process of sperm, which is the production of the eggs, and occurrence in the test is male. That means the production of the male, the spermogenesis. What is nothing but that is megasporogenesis and microsporogenesis, whatever the spermogenesis. But the spermogenesis is nothing but which production of the sperms. Okay, the next occurrence in the various in female, that is nothing but the which production of the female gametophyte. These are also very important, which means indirectly the meiosis process which are help in the reproduction process. Okay, and this is the common meiosis types. We are classified into mainly two different type of meiosis types which are present, that is meiosis 1 and meiosis 2. See, the meiosis 1 is nothing but that is the diploid cell. Just wait, I can show that. This is the mother cell which are having two set of chromosome here. This two set of chromosome which producing four cells. These four cells are belongs to haploid, and we have two different type of meiosis 1 and meiosis 2. And meiosis 1, which is nothing but the mother cell which producing two daughter cells, especially these two daughter cells are haploid cells. These haploid cells which producing which is participating again meiosis 2, and this meiosis 2 which producing again to two daughter cells, which means finally in the meiosis 2 we can observe 4 daughter cells. Is it clear? Next, so meiosis 1, this is also called as reduction division, and in first reduction division, which is having different type of stages like prophase and metaphase and anaphase and telophase. Okay, these are the developing stages which has occurred in. These are the most important stages which are present in the reduction division, either meiosis division, that is nothing but prophase 1 and metaphase 1 and anaphase 1 and telophase 1. See, why I am telling 1 is nothing but in the meiosis 2 we have again different type of substages are present, that is prophase 1, prophase 2 and metaphase 2, anaphase 2 and telophase 2. Orally meiosis reduction 1 or meiosis 2, I mean reduction 2, which occur in different type of 4 stages are present here. Okay, which means over here I am telling the stages. Then first one prophase, the prophase 1, which is having a proper nuclear nucleus, and nucleus is which is covering a proper nuclear wall, and in the inner side we can observe chromosomes. The chromosome pairs we have to identify. Okay, then metaphase. In metaphase, we can observe spindle fibers. We can observe both side dorsal and ventral poles, and we can observe different type of spindle fibers are there. In the anaphase, this is the anaphase, we can observe different type of chromosome shapes like V shape, like L shape, like J shape, like I shape. Okay, and next one, then last one, that is telophase, we can observe two different type of daughter cells are present. These are the reduction division which occur different type of stages like prophase and metaphase and anaphase and telophase. Most important. Next I am going to discuss about individually, that is the prophase 1. See, the prophase 1, this is also very important, and the prophase 1 which has having homologous pair, and which is occurring crossing over, and chromosomes onidae and spindle fibers and nuclear envelope fragments. Okay, before that, the prophase 1 which is having, you can see the diagram, the prophase 1 which are having the proper nuclear wall, and it's a very important. This is the early stage of prophase 1, and see, and in this prophase 1 which has observed pairing chromosomes in the lower side, and also a proper nucleolus is present here, and in the late phase the nucleus is completely absent. A nucleolus, see, we can observe in this one nucleolus is present here, and here nucleolus is absent. Just wait. And here also the nucleolus is absent. The chromosome pairing we can observe here, the chromosome pairing, and nucleolus is absent. This is the early prophase and this is the late prophase. Is it clear? In the prophase we can observe different type of substages is present, that is nothing but leptotin, and second substage is nothing but that is zygotin, third one that is poacetin, and fourth one diplotin, and then last, that is nothing but um, diakinesis. See, in the first I am going to discuss about leptotin. The leptotin, the nucleus is the prominent nucleus is present here, and also the chromosomes are very thin in nature. The chromosomes which has having and bulbous structures, these are called as chromomeres in the stage of leptotin. And next, the chromosomes are homologous chromosomes are present. in the first stage of leptotin. Now I am going to discuss about the zygote. The chromosomes are very short in size and which are showing like thin, and here also having homozygous chromosomes which are showing to one pair, that is called as bivalents. So that the type of reaction which is called as synopsis, either pairing system. Okay, this is which is present in zygote. Next, the third one, that is pocketin. The pocketin which are showing like this is the common synopsis, which is nothing but heterozygous chromosomes which has paired, that is called as tetrad. The tetrad which has present in four pairs and two chromosomes pairs are present here. Okay, next one, the packaging stage, we can observe bivalence, which means the paired chromosome, that is hetero. Next, these are the commonly which the homo and hetero chromosomes which is having two different type of sister chromosomes and non-sister chromatids. So that is nothing but a pocket in tetrads is present. These are the packet in tetrad. See, we can observe this is the packet in tetrad, which means which are having four pairs are present here. Okay, non-sister and sister chromatids are present. So non-sister is nothing but the chromatids are the extracted from the same chromosome. Non-sister is nothing but the chromatids are came from different type of chromosomes. Okay, the homologous pairs swaps pairs of chromosome and creates now combination of tetrads. See, this is the crossing over process, which is nothing but this is the two chromosomes which are showing like X-shaped structure, and this is X-shaped structure which is called as crossing over. Crossing over is nothing but non-sister and sister chromosome, the genetical material one chromosome to another chromosome, which means the one chromosome, this is second chromosome, the genetical material is transferred from one chromosomes to another chromosome. This is called as crossing over. Okay, this crossing over which is occurring pocketing stage, and the crossing over which are helping into enzymes especially, that is restriction endonuclease enzyme and ligase. What is the function of restriction endonuclease enzyme is nothing but this is first discovered by Nathan scientists. The restriction endonuclease enzymes are classified into 1 and 2 and 3, and restriction enzyme function is nothing but the cell wall of chromosome, I mean the rupture of the cell wall majorly. Okay, then ligase which has helping in the reconstruction of the wall of chromosome after the crossing over. What is the crossing over definition? The genetical material one chromosome to the transfer of genetical material one chromosome. See, this is the one chromosome to another chromosome majorly. This is crossing over. Okay, so in the diplotin and diakinesis, diplotin we can observe the terminalization process, and finally that is diakinesis, and diakinesis we can observe bivalences especially, and here we can observe in diakinesis, this is the lateral stage, we can observe different type of nuclear cell wall, and also nuclear wall, and also nucleolus, which also we can observe. Okay, now I am going to discuss about that is metaphase 1. The metaphase 1, the bivalent chromosomes which has preparing then metaplate in the middle of stage, that is called as metaplate, and we can observe two different type of folds like upper and lower side, and we can observe spindle fibers are also present here. The spindle fibers which is occur two different type of folds, especially this is the two different opposite poles. Just wait, I can show. This is the different type of folds we can observe like ventral and lower side. These are the spindle fibers we can observe, and these are the chromosomes which are present in the middle of metaplate. This is called as metaplate. Next, this is the anaphase. We can observe anaphase anaphase 1 usually. Then anaphase 1 is nothing but this. Here also we here also just I can show. Here also we can show two different type of poles, that is upper pole and lower pole. Then upper pole we can observe like this type of spindles are arranged like opposite manner, and each spindle fibers which are showing like a different type of chromosomes like this. The chromosome which are showing like two arms like this structure. Okay. uh, this type of chromosomes which are arranged in the central um, metaplate. Okay. So in the metaplate side we can observe like chromosomes, the chromosome which are showing like L shape. uh, just wait, like L shape and J shape and V shape, I shape. So these four different type of chromosomes means, that is metachromosome, submetachromosome, acrochromosome, telochromosomes. These are the main different type of chromosomes which is present in our body. This type of chromosome which can observe in anaphase, majorly anaphase, which are showing like anaphase like these structures in the spindle fibers which are having some space pressure, and the chromosomes are moved to the opposite pole sides. This is the opposite pole side. Okay. Homologous means after pressure the homologous separates and move to the opposite pole, and sister chromatids remains attached at their centromeres, these opposite poles which is called, that is dorsal and ventral, and each chromosome which are having like two arms, see like this structure, and this junction means this is called as centromere. Okay. This is called as primary construction. So now the last one, that is telophase. Telophase we can observe then same similar character of the prophase one initial stage, which means which are having a proper nuclear wall is present here, and inner side we have to observe a proper chromosomes, and which are observed, which means the nuclear envelope is present here, means resemble with the prophase 1 initial stage, and spindle fibers in the central side, and cytokinins which has occur, which is divided into two small cells. These are haploid. Why? Because here why I am showing haploid is nothing but the prophase 1 which has the genetical material, the transfer of genetical material, then one chromosome to another chromosome, which means the crossing over means the crossing over is nothing but the genetical material from chromosome to another chromosome, um, which is occur. Then only here which has producing two daughter cells, the daughter cells are having only haploid. Okay. This is the common review of meiosis 1. This is the common cell which are showing two set of chromosome here, they can mention here, and the then second one, the two set of chromosome double stranded, we can observe in double stranded here, and next one, the metaphase the double stranded, um, the chromosomes we can observe like red colored and blue colored, these are the non-sister chromosomes especially. And then finally, then we can observe different type of pairs, that is called as bivalent either synopsis, which has having non-sister chromosomes like red and blue, and one more and red and blue is present. The next one, this is just I can explain the now structure of the meiosis too, especially The second division, that meiosis 2, this is the meiosis 2 is a similar character, and the resemble with the mitosis 1, especially in this meiosis 2 is also having different type of stages, like four stages are present here, that is prophase 1 and metaphase 1 and anaphase and telophase. These are different type of the stages which is similar with the mitosis. Especially you can observe in the slide, that is the first one, that is that is double standard, that is meiosis 2 is, I'm telling meiosis 2, and we can observe which producing finally four cells. Is it clear? Next one, I'm going to discussing about the next stage. Just wait. This is the value of meiosis is nothing but meiosis introduce the genetic variation, especially, and gametes offering do not does have same genes as gametes of from formed from the parents. Especially before that time, with initial class I'm explaining about the meiosis process, which are helping in both microsporogenesis and megasporogenesis. That's a very very important. What is microsporogenesis? Megasporogenesis is nothing but the mother cell of megaspore, mother cell which producing four daughter cells, that is called as megasporogenesis. At the same time, microspore mother cell which producing four daughter cells, this is also called as microsporogenesis, which means indirectly and directly the meiosis which are help in the process of meiosis and sorry, in the process of sex organs, like male and female sex organs, like the formation of gems, and then formation of ovule and ovary. Okay, and this is a raw material for evaluation. Especially this is a mother and father, and this is a new gametes. Especially, okay, this is our meiosis. Again I'm explaining from starting onwards. These are the common fact of meiosis. The meiosis was first discovered JB Moore and Foreman scientist in the year of 1905. And meiosis is also called as reduction division. And in meiosis, which are having two different types, that is meiosis 1 and meiosis 2. And next the meiosis process, the cell which is participated only diploid cell, not haploid. Why? Because in this process the daughter cells which producing an identical structure, which means which are having only single set of chromosome, that is haploid. Okay, these are the dissimilar with the mother cell. Especially, and in the meiosis process which are having different type of stages, like prophase 1 and metaphase 1. This is the called as prophase 1. This is initial stage, and which are having early stage and later stage. And this type of prophase 1 again having subdivisions, like packetin stage, sorry, first stage, that is leptotin, zygotin and pocketin, and also diplotin, and finally that is diakinesis stage is there. Okay, and next one, this is the tetrad formation in the process of prophase 1. This is the crossing over which is present, which has occur in the particular substage of pocketin. And what is crossing over is nothing but the genetical material, one chromosome, the sorry, the transfer of genetical material one chromosome to another chromosome, that is called as crossing over. Especially this is occur at particular stage, that is x, the that is we can observe as x manner x stage, and this is called as crossing over. The crossing over occur whatever the two sister chromosomes, either non-sister chromosomes also. Hence hence we can observe in the end of meiosis anaphase, we can observe only haploid daughter cells. This is the metaphase, which can observe then spindle fibers are present here, and which can observe two opposite poles, like dorsal and ventral. And then the chromosomes are which occur in the middle, that is the middle, I mean plate, Okay, this is anaphase. Anaphase, we can observe different type of structures of chromosomes like L shape and J shape and V shape and I shape. These are common called as metaphase, or sorry, different type of chromosome types, that is metachromosome, submetachromosome, acrochromosome and telochromosome. These are which has present in, we can observe a specialized and proper structure in the anaphase only. Next, telophase. The telophase is nothing but the nuclear envelope is resembled and spindle fibers are present and cytokinesis which is occur. Okay, I mean then cells are divided into two. This is a common review. Again, I can show that is the first division of meiosis 1, and which can observe meiosis 1, especially the mother cell which producing two daughter cells. These two daughter cells are identical, non-identical with mother, which means the two set of chromosome, two set of chromosome cell is the two set of chromosome cell is producing haploid daughter cells. Okay, this is unidentical with the mother. This is meiosis 2, and this is the value of them which are helping in the process of sexual reproduction, especially the production of male gamete and female gamete, either in the formation of megasporogenesis and macrosporogenesis especially. Okay. um, Thank you, students, and thank you. This is my meiosis process. You can observe different type of stages, especially anaph sorry, first phase, prophase, and metaphase, anaphase, and telophase, and which can observe in the prophase we have different type of subphases are there. That is very very important for leptotin, zygotin, packaging, diakinesis, and diplotin. Okay, thank you, one and all.